Hello friends! A glorious, colorful evening to one and all. I hope you all are fine. Of course, we are always fine, happy and energetic. Let me introduce myself. I, 𝗬𝗧𝗻𝗂, pursuing 12th grade in BiPC stream from TTWREIS College of Excellence, Vegetative of respected Principal Sir M. Devadas Garu. My parents are Mr. Bhadru, Mrs. Sunita, and my mentors are Mrs. Vasanthakumari Madam and Mr. Krishna Sir. Today, we are going to discuss an interesting topic from the branch of Botany. But name of the topic is, it is in suspense. I am starting my topic with a small interesting story. After the completion of my story, you may come to know about my topic name. Let's start our session. Are you ready, my dear friends? Yes, so let's get started. Before starting our topic, what is the main beauty and attractive part of the plant? Well, flowering is a main beauty and attractive part of the plant, isn't it? Now, I'll tell you a story of same flowers of two friends, Pinky and Dinky. One day, Dinky gave a flowering plant to the Pinky on her birthday as a gift. They both planted in garden for their own to grow. After few days, they observed that the plant is having a change with growth of leaves and flowers. They enjoyed and also admired observing its growth. They saw its flowers are blooming on the trees and attracting all of us and also insects. They saw falling of leaves and flowers, and the flowers that remain become a fruit. That how a flower gives out a fruit? That how a flower gives out a fruit? Yes. To their surprise, they don't know how could it happen. So, they immediately run to their science teacher and post their amazing view that, teacher, my plant is having a change and it is finally differentiated into a colorful fruit. So how could it happen? Yes, the science teacher smiled and took them to their garden and explained that, what could be the answer of the teacher? Yes, it is due to the process of sexual reproduction. And what is our today's topic right now? Yes, today our topic is sexual reproduction in flowering plants. The sexual reproduction in flowering plants, the organ which helps in the sexual reproduction is the flower. We all know the various parts of the flower, right? A complete flower contains 4 floral parts: the calyx, corolla, androecium and gynoecium. Here, the calyx and corolla together called non-essential organs. Here, the calyx and corolla together called non-essential organs because they are indirectly involved in a sexual reproduction, where the androecium and gynoecium together called essential organs because they are directly involved in a sexual reproduction. Then, what is the male whorl of a flower? Right, androecium is a male whorl of a flower. Do you know which parts unite to form androecium? Yes, stamen unite to form androecium. So, let's discuss about the structure of stamen. The stamen is a flowering part. The stamen is a flowering part that contains pollen grains. How see the pollen grain is? How see the stamen is? It contains 2 main parts. The long, slender and stalk-like portion is called filament, where the fertile portion of the stamen is called anther. How see the anther is? The anther is bilobed, with each lobe having 2 thecas or 2 sacs. Based on the internal structure and the number of thecas present in the anther, The anthers are of commonly different types, monothecous anther and dithecous anther. We know that single-lobed anther is called monothecous anther. We'll see the monothecous anther in Malvaceae family. For example, hibiscus, cotton, okra and etc. Where two-lobed anther is called dithecous anther. We'll see the dithecous anther in Solanaceae family. For example, datura, tomato, chili and brinjal. So friends, if we see the transverse section of bilobed anther, the anthers are having four-sided tetragonal structure consisting of 4 microsporangium located at the corners of 2 in each lobe. So let's see our activity of bilobed anther under microscope. What is our aim today? Today our aim is observation of bilobed anther under microscope by section cutting. To see the bilobed anther under microscope, the material required are microscope, plant of a bilobed anther, slides, watch glass, cover slip, brush, blade, glycerin, safranin and water. In this procedure, first I am taking 2 slides and pour water in first watch glass. Pour water in first watch glass and pour water along with a drop of safranin in second watch glass. Now, I am taking a plant of bilobed anther and slowly cut it up to get a thin layer. And slowly cut it up to get a thin layer. And place it in slide that we have added water in it and transport it into another watch glass that we have added safranin. Now I am taking these pieces into water and now I am taking a slide. Now I am taking a clean slide. I am choosing a thin layer of bilobed anther so that we can see clearly under microscope. Now I am adding a drop of glycerin on the slide and I am keeping cover slip on the slide slowly. And slowly I am keeping cover slip on the slide so that our activity is ready to observe under microscope. I can observe bilobed anther under microscope and after my observation I can see clearly the bilobed anther. So friends, in this bilobed anther, a single lobe contains 2 microsporangium and 2 lobe contain 4 microsporangium. So let's discuss the detailed structure of microsporangium. We can see clearly the bilobed anther under microscope. The microsporangium is a typical structure consisting of 4 wall layers and generally it is surrounded by 4 wall layers: epidermis, endothecium, middle layer and tapetum. Let me know you the structure of each layer. Let's go with first layer, epidermis. Epidermis is outermost wall layer which is made up of single layer of flat cells. It is a one-celled thick and the cells present between the pollen sacs or thin wall, and this region is called as stomium, which is going to help in the dehiscence of pollen sacs. Another layer is called endothecium. Endothecium is second outermost wall layer present just below the epidermis, and it is also made up of single layer, but they are compactly arranged with cellulose fibrous thickening, which is made up of pectin and lignin. The cells of this layer are callose thickening, and at maturity, these cells lose water and going to help in the dehiscence of pollen sacs. Underneath this layer, the cells from 1 to 3 layers form middle layer, which is made up of thin wall parenchymatous cells. The cells of this layer are ephemerals. So friends, these outer three most layer perform the function of protection and going to help in the dehiscence of pollen sacs. At maturity, these cells lose water and become fibrous and has rigid wall. Another layer is called tapetum. The tapetum is innermost wall layer, which is a special layer. It is going to provide many important functions. The cells of tapetum has dense cytoplasm and contains more than one nucleus. Can you think, friends, of how tapetal cells could become binucleated? Let's see. The cells of the tapetum undergo endomitosis, where nucleus in the membrane splits but not the cytoplasm. It means karyokinesis occurs in a membrane but not the cytokinesis. If we see the function of tapetum, it is a nutritive layer which is going to provide nutrition to the developing microspores. The cells that secretes the callose thickening, that means callase enzyme. It dissolves callose of microspore tetrad. The tapetal cells also provide 𝗂𝗍𝗃 bodies which is coated with sporopollenin used for the development of pollen. So friends, as the anther is young, the group of compactly arranged homogeneous cells is called sporogenous tissue, which occupies at the center of each microsporangium. As the anther develops, the cells of the sporogenous tissue undergo meiosis division and form 4 haploid microspore tetrads. This process is known as Microsporogenesis. As we all know that each cell of the sporogenous tissue is also called pollen mother cell or microspore mother cell. These undergo reduction division and form 4 haploid microspore tetrad. This process is termed as Microsporogenesis. Here, the microspore tetrads are haploid in condition and the pollen mother cell are diploid in condition. The microspores formed are arranged in a cluster of 4 cells and together called microspore tetrad. These microspore tetrad are of different types: tetrahedral type, isobilateral, linear, T-shaped and decussate. Out of these, the tetrahedral type is most common tetrad which is present in the cells of the tapetum. So friends, as the anther get mature and dehydrated, the microspores dissociate from each other and develop into pollen grain. So let's see what actually the pollen grain is. Pollen grain. The study of pollen grain is called palynology. The study of pollen grain is called palynology. It represents male gametophyte. The formation of pollen grain from microspores is termed as microgametogenesis. Here, microspore is also haploid and pollen grain is also haploid. This process is termed as microgametogenesis. Have you seen hibiscus? Absolutely. If you touch the open anthers of the hibiscus, you will find a large deposition of yellowish pollen powdery that actually the pollen grain. The pollen grain has 2 prominent wall layers, the exine and intine. The exine is outer hard layer. It is irregular and discontinuous layer which is made up of sporopollenin. The sporopollenin is one of the most material resistant organic material in the world and no enzymes that degrades the sporopollenin. The extant pollen grain has a prominent aperture is called germpore and where it is present where the sporopollenin is absent. The germpore is surrounded by a hole on the microspore, it means a pollen grain. It emerges during germination as a pollen tube. The inner layer is called entyne. The entyne is thin and continuous layer and it is made up of pectin or cellulose or pectocellulose. The cytoplasm of the pollen grain is surrounded by plasma membrane. At maturity, the microspore nucleus undergo karyokinesis and produces two nuclei and the separation of cytoplasm. It means cytokinesis is not equal, that it has one larger cell and one smaller cell. That actually the larger cell is called vegetative cell and smaller cell is called generative cell. And what these cells? Let's see. The vegetative cell is bigger and has abundant food reserve in it. It has an irregular nucleus, but the vegetative cell is not going to help in the formation of male gametes as it is non-reproductive, where generative cell is smaller cell and floats in the cytoplasm of the vegetative cell and it has dense cytoplasm and a nucleus and a spindle-shaped cell. In ova, the 60% of the angiosperms released from the anther at two-celled stage, the vegetative cell and generative cell. Remaining 40% of the pollen grains are released from the anther at three-celled stage, the vegetative cell and two male gametes that are divided mitotically from the generative cell. So friends, we all know about pollen grain, but how many of you know the uses and harms of pollen grain? Let's see the advantages of pollen grain. The pollen grain is essential for sexual reproduction in flowering plants. It is a nutritive and become very fashion to use pollen tablets as food supplement. If you see in Western countries, the pollen products are in the form of pollen tablets or pollen syrups that are available in the market. The pollen consumption has been claimed to increase the performance of athletes and racehorse. If we see the disadvantages of pollen grain, the pollen grain of large number of species causes a severe allergies. In some people, this often leads to chronic respiratory disorders like asthma and bronchitis. The contamination and the inflammation of pollen allergy by the pollen grain is due to Parthenium hysteroporus, which is available in the India. But it is possible to store in liquid nitrogen at -196 degrees centigrade as pollen banks similar to seed banks in crop breeding program. So friends, let us generalize our topic once again with mind mapping for our better understanding, right? Flower. Flower is a site of sexual reproduction. It is composed of 4 whorls, calyx, corolla, androecium and gynoecium. Out of these whorls, androecium is male reproductive organ which is composed of stamens. The stamen contains 2 main parts, the filament and anther. The anther is fertile portion of the stamen. It contains 2 lobes and each lobe contains 2 sacs or 2 thecas and each theca contains a microsporangium. The microsporangium appears circular in its outline and composed of 4 wall layers, epidermis, endothecium, middle layer and tapetum. The outer 3 wall layers perform the function of protection and going to help in the dehiscence of pollen sacs. The tapetum is nutritive layer going to provide nutrition to the developing pollen grain. Inside the tapetum, the cells of the sporogenous tissue undergo reduction division and form 4 haploid microspore tetrad. And these haploid microspore tetrad undergo equational division and develop into pollen grain. The pollen grain represents male gametophyte. It contains 2 cells, vegetative cell and generative cell. The pollen grain is also used as advantageous as it is nutritive and also disadvantageous as it causes severe allergies for human life. But it is possible to store in liquid nitrogen at -196 degrees centigrade. So friends, now the Pinky and Dinky both understood clearly that why their plant is having a change with growth of leaves, flowers and fruits. That each flower gives out a fruit, through sexual reproduction. Yes, now we'll again meet in the next session that they both went to their home happily, and for us also it's time to off the session. Now I am very thankful to our Telangana government and our respected Secretary Sir Ronald Garu, and my special thanks to my principal Sir M. Devadas Garu, and we'll again meet in the next session with another interesting topic, structure of gynoecium, and don't miss it. Thank you one and all. Jai Bhim.